Okay, so we have switched to English. So for the non-French speakers, I will follow in English, and you can, of course, follow my slides. They are written in French. So I will talk today about my main focus of research since many years is the study of repair of oxidative DNA damage, and I'm particularly interested in two approaches, in vitro and live cell imaging approaches. So I'm working in the IAB in Grenoble, as mentioned. So DNA repair is essential for life. Every day, in every single cell of our body, we have more than 10,000 lesions that are occurring from different sources of either exogenous sources or endogenous sources, but we can repair it. But we know that in absence of a complete repair of this DNA, as in some rare genetic disease like xeroderma pigmentosum, you have a dramatic increase in skin cancer. We have also other syndromes that are more complex, but like the cocaine syndrome. And you will have, in this case, a segmental premature aging. And there are more and more evidence that all the multifactorial diseases related to age, such as cancer, neurodegeneration, but also diabetes, are all connected to a deficiency in DNA repair. And we are studying this to understand better how DNA repair occurs. I will just briefly introduce the two main excision repair pathways. So on the left is the base excision repair, dealing with non-helix distorting lesions, such as 8-oxoguanine. They are removed only one nucleotide by a glycosylase called OGG1. On the right, you have the nucleotide excision repair pathway, which is dealing with more helix distorting lesions, such as UV lesions. And they will remove a patch of 25 nucleotides, and it's initiated mainly by XPC and CSB proteins. So there was some methods developed over the years to study DNA repair pathways, and especially the nucleotide excision repair, but also double strand break repair that you have heard about it, certainly, such as gamma H2AX. But there was no satisfying tools to study the base excision repair, at least locally in cells. And that's what I've been developing during my stay in Erasmus Medical Center in Rotterdam. And I'm now establishing this method in IAB. And I won't go in detail of this method, but this allows us to to generate locally 8-oxoguanine. And you can see the recruitment of the glycosylase here in this region. And we do that without other generation of double strand break or UV lesion. And so we can study base excision repair without interference. And this led me to discover a new interaction between NER and BER, as you can see here. And what was even more interesting is that in two cells co-expressing this factor XPC fused to the fluorescence protein cherry, or a CSB fused to a GFP protein, you can see that the recruitment vary depending on the transcriptional status of the DNA. And we are now studying this in more details, and we want to understand how the conflict between transcription and reparation of the DNA are dealt by the cells. In physiological conditions, like in the presence of CSB, you can have a complete BER, but our current model of studies is relying on this model that in absence of a protein such as CSB, you will have a cut in the DNA that will then stop the polymerase generated by the glycosylase OGG1, but this will lead to an incomplete repair, and this will be even more detrimental and stop transcription and full reparation. And we are currently exploring in more detail this model. As mentioned, I'm studying also in vitro this aspect, and as you all know, our DNA is not naked in the cells, but it's in the form of nucleosomes to wrap the 2 meter of DNA in a few micrometer cells. And I've been developing such a tool where you have a single 8-oxoguanine in the context of a nucleosome. So we've been showing that ATP-dependent chromatin remodeling was necessary to access these lesions, and also a removal of another histone linker such as H1 was necessary for there. to occur. But we are now moving to the next step of folding of the DNA, which is the so-called 30 nanometer chromatin structure. And you can see here that thanks to our latest collaborations with people in IBS Grenoble, we have been able to get a very detailed structure of 30 nanometer chromatin. And now we can include lesions in different positions and study how this lesion will be repaired. And the ultimate goal of our group is to determine by cryo-electromicroscopy the structure of this complex. And this was indeed my last slide. I have to thank the people working with us, especially all our collaborators. This is, of course, a collaborative work. And I thank you all for your attention. Merci.
Merci Hervé. Du coup, je me demandais au sein de l'IAB, est-ce que tu as développé une plateforme particulière qui est accessible à la collaboration pour étudier ce type de modèle?
Dans le sens de l'étude sur des cellules vivantes?
Oui.
Oui, bien sûr, c'est ce qu'on développe actuellement. On a donc c'est sur une plateforme de microscopie confocale sur laquelle on peut étudier des cellules vivantes et voir dans différents contextes avec des inhibiteurs partiels, avec des cellules KO pour certaines cellules. On peut étudier les recrutements et mesurer très précisément en fait une influence sur la réparation de l'ADN.
Très bien. Et ben pour terminer les questions, une question de Céline Hadji qui travaille dans la société Cell & Soft. Les essais in cellulo sont-ils réalisés sur des plaques de culture classiques ou sur des substrats spécifiques?
À l'heure actuelle, on utilise des lignées qui poussent sur des cultures classiques. On peut passer éventuellement sur des cultures 3D, mais on a d'autres inconvénients parce que en microscopie confocale, c'est pas évident de travailler sur un contexte 3D pour le moment, mais on développe ça aussi.
Bon, bah, très bien. Je pense qu'il y aura peut-être matière à interagir et peut-être solliciter un rendez-vous entre vous deux. Merci encore Hervé pour la patience.
Merci à vous. Je savais pas quand j'allais passer.
Un an plus 5 minutes. Voilà. Bon.
Merci.